Today I decided to talk about clinical trials while we're talking about basically because it's relevant to COVID and it's relevant to vaccines. But before I do that, I just want a PSA. Since Chicago is reopening, we are now in phase 3 reopening. And I have seen a lot of people I live right in the downtown area, so there have been a lot of people here of late not wearing masks properly. So the whole point is to prevent respiratory droplets from leaving your body, and your proximity. So it has to cover your nose. Yes, Blanca is demonstrating the correct way. And so it has to cover your nose. And the reason it's important that it comes all the way down over your chin is that they can escape down that way. And it's a little uncomfortable, but as someone who has been wearing a mask all day, every day for almost 2 months now, you will eventually get used to it, and your skin will eventually get used to it. Wash your face.
It's
Okay.
Frida Oh, sorry. Monica asked what
I
it is.
love it.
Frida Kahlo.
That's cute.
That's awesome. Okay, so, and this is just because I think that there's a lot of confusion about this. So does anyone know, who does wearing a face covering protect? Does it protect the person wearing it? Or does it protect other people? Oh wait, I can't open it.
Saying others.
Yeah, you got
in the
others.
chat.
So.
Yeah. So that's correct. And I think that this is important because I've seen a lot of social media posts about not wanting to wear a face mask, and like people who aren't afraid to get COVID not wearing a face mask, but it's not really about you, it's about protecting other people. Yeah, so like the vast majority of what a face covering is doing is it's preventing your respiratory droplets from getting everywhere. But if we all wear them, then we can protect each other. And wouldn't that be so nice? So if someone tells you that they don't want to wear a face mask, remind them that it's not for their safety, it's for other people. And then the last thing I wanted to address is that in the last 24 hours, the WHO has announced, I don't know if anyone else is following this, but WHO announced yesterday that asymptomatic carriers were not transmitting. And then today announced that 40% of asymptomatic carriers are transmitting the virus. And the reason I just want to bring that up is that, again, I've seen a lot of people who are trying to listen to these big organizations and are getting confused. And, it's a new virus. So as information comes to light, some things are going to change. But as of right now, as of today, as far as we can tell with all the contact tracing studies, people who are asymptomatic can spread the virus. And so even if you have no symptoms, it's still important to maintain social distancing, wear masks, and in general, try to protect the health of yourself and others, especially if you're around people who are older, who are more likely to get very sick from this. So that's just a PSA. And I just want to give you guys the information because I feel like the point of having information is to share it. So now you have been deputized to teach others about social distancing. Okay, so the first thing that I want to talk about is, so they're developing a successful vaccine against SARS-CoV-2, which is the virus that causes COVID-19. This is blocked out because it's really messy and we'll look at it in a second. But these are basically the different, can you see my pointer? Oh yeah, here we go. These are the different strategies used for a vaccine. So live attenuated, and they're kind of in order of, well, we'll talk about that in a little bit. But so a live attenuated vaccine is a vaccine where you take the entire virus and you heat or chemically treat it so that it is not as viable. But then you inject the whole virus like that into someone. And then there's viral vector-based vaccines, which means they can produce the proteins for the outer portion of the virus, so you get the entire capsid of the virus in your body to make antibodies against. And then recombinant protein-based vaccines, in this case, it's the spike protein, which we're going to talk a lot about, which is, if you've seen the picture, it looks like a spiky ball. It's that spike. This was not creatively named. And so these vaccines, what they do is they either give you a high dose of this protein specifically, or they give you a way, they give your body a way to make the protein either with DNA or mRNA directly. And so exposure to a high amount of a foreign protein in your blood will create an immune response. Does anyone know what that sounds like? An immune response against just like a protein that isn't doing anything? So normally, if this happened, it would be considered an allergy. So this is the same mechanism by which you can be allergic to peanut butter or to pollen. There are proteins that your body is recognizing as foreign. And so you have this like inflammatory response to those. And so they're in this order, the vaccines. These vaccines, the live attenuated vaccines, are the most dangerous in terms of the most likely to actually cause disease. And the mRNA vaccines are the least likely to cause disease. However, the mRNA vaccine is also the least likely to cause robust immunity. And the live attenuated vaccine is the most likely to cause robust immunity for the same reason. So the mRNA vaccines are the ones that we'll focus on a little bit coming up. And so basically, do you guys know what mRNA is?
Put down the chat.
No?
Does anyone know what mRNA
Yeah, it's here.
is?
Okay.
Messenger RNA.
Messenger RNA. Yeah. So messenger RNA. What does messenger RNA do? What's its job? Maybe that's a bad way to ask the question. Yeah, sends a copy to ribosomes. That's great. So basically, what you guys are getting at, to make the proteins. Awesome. Yes, perfect. Yeah, so it's called a messenger RNA because it's the messenger and it carries the information to make a protein. So the reason this is safe is also that messenger RNA is pretty easy to degrade and that it's only going to make one protein. It's not going to make a whole virus. And so mRNA vaccines are the vaccines that are being focused or mostly made, but we'll talk about that in a minute. So this is the rest of this. So this is we're not going to go through this whole slide because I think it's really messy. But I wanted to have everything on here so that you see that this is a really complicated system that we're putting foreign bodies into. So here is whatever is in the vaccine, and it activates this whole massive immune cascade. And the end goal, ultimately, is to create these neutralizing antibodies. And so that's why it's important to make something like the spike protein or the capsid of the virus that antibodies could recognize, because the whole idea is to knock down the virus before it even enters cells to replicate. So that's kind of how a vaccine works. And these are the COVID vaccines that are currently in process. Like I said, so the spike protein is this little red guy that's sort of spiking off the capsid. So I have them separated into governmental agencies and private corporations and what they're doing. And you can see the vast majority of them, I kind of listed, are mRNA viruses. And so this mRNA vaccines, and this mRNA codes for the spike protein. So it's just going to give the body the tool to make this foreign protein and then essentially cause an allergy equivalent to that protein. But in this case, it's going to look like antibodies against that protein. So hopefully if your body sees that protein attached to the viral capsid, then it will also be able to kill the virus. So because mRNA is easily degraded by the body and the mRNA coding for a foreign protein, especially, and that this is not a virus in the sense that it cannot invade cells, it cannot replicate in cells, cannot lyse cells. So in the NIH is working on a trial in Seattle that has actually skipped its preclinical phases and is going right for just injecting this vaccine into healthy volunteers. And so we'll talk a little bit about what that means, but this is in an effort to speed up COVID vaccine generation. You may have heard that the EMA is also, making an mRNA-based vaccine. And then in Australia, they are also skipping the preclinical phases for that vaccine. But in Australia, they are not skipping the preclinical phases. They are testing it in ferrets because ferrets contract the ferret coronavirus the way that humans contract our coronavirus. So they're a really good model system, which is what I learned. They cough on each other and inhale it and then get coronavirus. Who knew? And then Noravax is a private company that's actually partnering with Australia to do to do these trials. So the private corporations are trying something a little bit different. Sanofi and GSK are making an S protein COVID-19 antigen. So they're actually making this protein. And then they have an adjuvant, which means it's added to another protein that's known to cause an immune response. So this type of vaccine will cause a very strong immune response and is likely to trigger immune memory and make those antibodies last a really long time, or at least make cells that can make those antibodies that will last a really long time. But for these ones, obviously, these are being trialed in animals first because of safety concerns, since injecting a large dose of a protein obviously can be very dangerous. And then Pfizer and BioNTech are making another mRNA virus, and then CanSino in China has made a protein. This protein actually transfers the spike. So they will inject spike proteins and this protein, which will transfer just the spike into cells, and the cells won't like it because they're foreign, and then induce an immune response that way. And this protein all on its own has no effect. Okay, so those are the vaccines that are happening right now. I don't know if anyone has any questions about the vaccines that people are working on. Okay. So the rest of this is really to explain why, I have a lot of friends who did not go into science or medicine, and I am often asked why we can't have a vaccine really fast. And so this is kind of why, is the process of going through clinical trials and FDA approval. So the steps here are designed for drug trials, but the same steps are actually taken for a vaccine because it's still a foreign substance that you're putting in someone's body. So the preclinical phase is the phase with animals. Like I said, in Australia, they're using ferrets. And really, this is to look at the mechanism of whatever you're studying, safety testing, whether it's effective at all. And secretly, there is usually a calculation to find out if it would be lucrative. So if there are things that work that wouldn't make it past the preclinical stages if it's not worth going through all of the next stages for a company. So for very rare diseases, for example, this is a major issue in terms of getting treatments made. Can anyone think about any type of safety or toxicity testing that you couldn't do on people that would be unethical to do on a person? Or if there's a certain type of person that you can't do drug tests on?
Hey guys, anyone in the chat? So can you think of any group of people or
The elderly? That's a good guess.
who put a link in the chat? Are we supposed to go to the link?
Children, you're getting closer. So the big one that you really can't, is totally unethical, is safety testing on pregnant women. So we test on pregnant animals to see if drugs or vaccines cause birth defects or fatalities of fetuses, because it's considered completely unethical to test on pregnant women. Children and elderly, children in general, are not going to show up until phase 4 because they're minors, and unless something is specifically designed for children. And the elderly as well will show up, but only if it's important that it be tested on people who are older. So you're right that there are other populations that we don't like testing on. But yeah, so we do a lot of safety testing here. And in general, the most time is taken up with the preclinical phases of a drug or a vaccine. So like I said, the NIH is currently skipping this phase entirely and just taking that coding sequence mRNA and starting right with healthy volunteers. So phase 0 of a drug trial or a vaccine trial is often skipped or combined with phase 1. So it has 10 to 15 healthy participants. And the main point of this is to look at the pharmacokinetics. So that means how long does this stay in the body? And at what point does it reach peak dose in the blood? And in the case of the vaccine, the big thing that they're looking for is, do people make antibodies? And if so, how long are those antibodies stable? Can we, you think the anti-vax population? Yes. So I'm gonna get to that at the end, but yes, that is probably going to be a problem in this country. So in phase 0, it's a really small dose of the drug or a really small dose of the vaccine. And it's open label, which means that all participants know exactly what's going in their body. And the person administering it also knows exactly what's going in their body. So phase 1, are different doses safe? Oh, and sorry, just one point that I wanted to make is, like, notice how many of these phases we go through before we even care if the drug works. So because a lot of the FDA is just to make sure that people are safe. We don't want people taking or putting anything in their body that is unsafe. Okay, so are different doses safe? So this is going to be 15 to 30 people, and you're looking for dose response curves. So this is people who will get different doses, in this case of the vaccine, to look for immune response. Again, this is healthy volunteers, and again, this is open label. Everyone knows exactly what's going in to these volunteers' bodies. Phase 2 is we start to ask, does it work, and is it safe when it's working? So now we're getting into the hundreds of people. This is the first phase at which people with the disease that we care to treat will be present. This is the first phase also where you may try it in a combination treatment instead of on its own to see if it's safe in combination with other drugs. And this is the first phase at which blinding may be introduced. There's usually 2 phase 2 trials done, one that's open label and one with blinding. So blinding means that people know they could be taking a drug or not. And it's really important to determine efficacy of a treatment. But the reason the previous phases were open label is just to protect people, because until we're 100% sure that it's safe, we don't want to be giving it to people who may or may not know that they've taken it. Phase 3, okay, so just to calculate, so we have tested on up to 600 people in phase 2, up to 30 people in phase 1, up to 15 people in phase 0. So we are now at 645 people have already tried this drug, and now we're looking at how does it compare to the standard of care. So these phase 3 trials is what you may have heard about when you hear about drug trials. They're much larger, and there is a control group. So these are the trials that are randomized, so people know that they could be taking one of 2 treatments. or they may have more than 2 groups if there's more than one standard of care. And we're really looking for efficacy and off-target effects. So you may have heard of the placebo effect, but for the most part, phase 3 trials are actually done comparing to what we would give people anyway. So if there is no standard of care, then we give people a placebo. But if there is a standard of care, there will usually be 1 placebo arm and 1 standard of care arm, and then 1 of the new drug, because you want to see not only that it works, but does it work as well as what we already have? And that has come to matter more than if it works. And then off-target effects, so that's side effects. Because sometimes you have 2 drugs that have the same efficacy, but they have different side effects, so you can give them to different people. Okay, so phase 4 is post-marketing surveillance. So after phase 3, if a drug passes phase 3, which most don't, 90% of drugs have been eliminated at this point. That started clinical trials. 90% of drugs that start clinical trials will already have been eliminated at the point that they get to phase 3. So if you pass phase 3, you go into phase 4, which is called post-marketing surveillance. So this is after the approval of the drug. So now we're looking at hundreds of thousands of people. We can look for rare side effects. We can look in combinations with other drugs, but it's still considered part of the trial, with the idea being that the FDA can at any point decide that this drug is unsafe and take away its approval. So what does this have to do with the vaccines? Like I said, right now we're still in healthy participants who know the dosage that they're getting, and they're going to look for if they develop antibodies and for how long those antibodies stay. And then as they move into larger and larger groups, what we're going to see more and more of are side effects of the vaccine and whether or not that immunity is generated in all groups. So that's the end. So I'll respond to the anti-vax thing, and then if anyone else has any questions or comments, please pop it in the chat. But so in general, in the United States, adults are less vaccinated than in other developed nations, like especially in Europe. And what phase are we in for? So right now, for all vaccines that are in phases, they're in phase 1. If they're in people, they're either preclinical or they're in phase 1 if they're in people. So in terms of, okay, so first of all, I mean, I'm going into healthcare, obviously, and I would consider myself pro-vaccine. Vaccines in general are lower risk than getting the disease that they're vaccinating you from. And if you go on the internet, you can find plenty of horror stories from when we didn't understand as much about the immune system. But all of that information is taken into account when they design vaccines. For example, the reason they feel comfortable skipping the preclinical trials in the United States is that they're using an mRNA vaccine, which cannot, by any stretch, turn into anything that's infective or lethal. And so, and we know that because of all the science that has come since, for example, the polio vaccine that was such an issue before. That was a live attenuated vaccine, and some of them were not sufficiently attenuated, and so people were just getting polio. And so when you read about that, it's easy to see why people are afraid of vaccines, but in reality, all vaccines that are recommended by your doctor are less dangerous, usually by powers of 10, so like 10 times less dangerous or 100 times less dangerous than getting the disease. The second sort of thing with vaccines, which is that a lot of the anti-vax movement in the United States started with someone who claimed that autism was related to MMR vaccines. MMR is measles, mumps, and rubella. It's a vaccine that you get as an infant. All three of these can be lethal to infants, which is why you get it. And so, first of all, since then, several 100,000 children with autism and several 100,000 controls have been studied, and no correlation was ever found between any vaccine of any type or any preservative used in a vaccine and autism, or actually any other neurological issue. Yeah, so the original study was not simply bad science, it was actually completely debunked. That study was fraudulent. So that man is actually living in the United States because there is a warrant in the UK because he is considered to have done harm to any child that was not vaccinated because someone believed his study. So that fear is unfortunately misguided, and it's been misled, and it's hard, right? You know, scientists are supposed to be honest and caring and trying to do the best they can, and for the most part they are, and this one just gained a lot of traction and it was wrong. So that is actually not true. And then the last thing with vaccines, something that I hear a lot is that people don't want, for example, the flu shot, or there's a pneumonia vaccine when people get older, because you sometimes get symptoms of the flu after getting the flu vaccine. So the flu shot is actually a live attenuated vaccine, which is why you have symptoms. However, if you actually get the real flu following the flu vaccine, you are significantly less likely to get terrible complications. And now you guys are all young and probably healthy, so you may not know a lot of people who got the flu and then got pneumonia, certainly not your age. But building up immunity over time, because the flu vaccine changes every year, will build up a giant library of immunities and be better prepared for any new flu. So that's one thing. And the second thing is, like I said, you are significantly less likely to get meningitis, which is an infection of the spine and brain fluid, or meninges, which are the capsules, I guess, of the spinal cord and brain. Yeah, and so you're going to get meningitis, and you're not going to get pneumonia if you get the flu. And so that's a big thing with like the flu shot specifically, is that yes, you may get symptoms from the vaccine, but it will not make you sick. And then later, if you get the real flu that's very virulent, you're significantly less likely to get these really extreme complications. So that's that.
I can share on that as well. I mean, I work in a pediatrics clinic in Austin. We're on the west side. And we had a kid die from the flu. There's a 15-year-old kid, did not get the flu shot and died because of the flu. So it's not entirely preventable. The flu vaccine is not perfect. But I mean, that's a terrible tragedy that really doesn't have to happen. It's very rare to die from the flu, but it's also extraordinarily rare to have really serious side effects to the vaccine. And certainly in clinic, I can say that a large number of parents refuse vaccines, and I think that's a big challenge.
Just another issue to point out. So a reason why this can be a big social issue is because of the concept of herd immunity. So you may have heard of this in the news as well, but for certain populations, vaccines don't work because either their immune system, they were born with an immune system that just had some defect, so they may not really have the capacity to build an immune response at all. So those people will always be at risk. And the reason why there's this concept of herd immunity is that if the majority of the population is immune to a certain virus, that virus kind of has nowhere to go. and it can't really exist in the environment. So then you decrease the chance that a person who didn't respond, who cannot respond to a vaccine, so you decrease the chance of that person contracting the disease because the disease is no longer around in the environment. So that's another to think about when it comes to vaccinations.
And you've probably seen, you may have seen for COVID, we are absolutely nowhere near the infection rates to get herd immunity, assuming that we even get a strong immune response that's durable in our society. I mean, the estimates are that we need, I think, the lowest I've ever seen is like in the 50 or 60% of the population has to be immune. And right now we're definitely less than 1%. So herd immunity is not a factor at this moment.
Right. For COVID, it's definitely not really relevant right now. But if you think about something like measles, like you've probably already seen outbreaks of measles in the news, and that's a vaccine that's just like given to every child, or at least offered to every child at birth. Even just a few non-vaxxers can lead to this popping up of measles cases in the community. This might be a bit sensitive, but will the protests spike cases?
I think the general consensus is yes. I think it's always a balance between personal safety and really doing what you believe in. So obviously we're not protesting, as safe as you can. Anytime you have a risk of the coronavirus, the advantage is that they're outdoors. Certainly, I think that there will be a spike.
Something that's important to know, there are protests that are sort of left alone, and so when people are people and everybody's wearing a mask, it's going to be less of an issue.
Any other questions for Sage? I'm going to stop your screen share, Sage, so I can put mine up. Thanks. All right.
Question.
All right.
And yes, we can definitely answer questions not about COVID. And this is
No
somewhat
more
related.
questions. Sorry, I put this up
about,
already.
yeah,
But
no worries. Can we
we
talk about
can
MIS?
hold off on my stuff.
Wait, what is MISC? I've never heard of that.
Yeah, you guys are welcome to ask us any questions you want. Obviously, Blanca will tell us if we are being inappropriate, but I don't think we're veering into that territory yet.
Multi-system inflammatory. Oh, it's the Kawasaki response to COVID.
Yeah, I haven't seen so much about it lately. I haven't been following it
Gotcha.
closely.
Yeah.
Okay, so for that, I mean, I think we talked a little bit about, yeah.
Yeah, there were cases in New York earlier.
We talked a little about why a vaccine, okay, hold on. All right, so if a person who is COVID positive coughs into the air, how long does it stay?
That depends.
Okay, into the air is like, how long does it stay in that air? Like if they coughed into the air and then walked away and then you walked through that air. I mean, in general, on surfaces, 3 to 5 hours is the estimated amount of time that a fomite, which is like a surface that got coughed onto, will carry that disease, will carry COVID or this particular disease. If they cough onto you, that's a little bit different, or if they cough in your direction, move away. But yeah, 3 to 5 hours on surfaces. So
I think this is also part of the reason why we have a 6-foot distancing rule, because in general, and this is very difficult to establish this, but in general with other diseases that spread with a similar mechanism to how we believe COVID spreads, these respiratory droplets, which have been modeled, and you can make droplets, tend to sink to the ground under the influence of gravity within 6 feet-ish. And there's another important concept here, which is if you're exposed to one virion or one particle of the virus, it's unlikely that you would have an overwhelming infection in response to that. But if you are exposed, like say if you're an anesthesiologist who's intubating a patient and you're just right in their mouth and they're just breathing straight on you, then you may be exposed to millions or billions of these particles. So there's definitely a response that varies with the dose response. So the more coronavirus particles you're exposed to, or the more viruses you're exposed to, the more likely you are to come down with floor disease.
So, all right. Let's see. The MIS-C and okay, so if someone had COVID, are they immune to it after they don't have it anymore? So that's actually what people are debating really heavily right now. In principle, yes. And that has to do with the way that you make antibodies is to generate a group of cells that will remember a foreign substance and remember how to make the antibodies against it. So that's how you maintain immunity. And right now, there's a lot of debate in the literature about how strong the immune response against COVID is to begin with. But theoretically, which is why countries that were thinking about releasing, what do they call them? Like health passports, ultimately didn't, because it's not 100% certain at this point whether or not having had COVID and gotten over it means that you're sufficiently immune to not get it again and get sick. So the MIS-C, I think the best way to think about this is we talked a little bit about why giving someone a vaccine might cause an inflammatory response like an allergy. And it's sort of the same idea, which is that, and we also talked about ARDS, I think, previously.
We talked about
yeah.
ARDS, yeah.
Didn't we on the first day?
Maybe for a second,
anyway,
but yeah.
Yeah, but so I think that the general idea is that these children are having a very, very extreme immune response to the virus itself. And so that's why they get this disease. I don't know what else you want to know in terms of like, I don't know if there's a good way to predict who's likely to get it or anything like that. As far as I have seen, it's just something that now we know if a child tests positive for COVID to watch out for early symptoms so that we can treat it before they get really sick. Okay. Investigating the best way to cover your mental health after a disaster. Is there a good, ethical, plausible way to measure one's mental health? Yes, there are. I think what I would recommend, you guys can jump in, is there are standardized questionnaires to measure like depression and anxiety scales. And if you can do that in a de-identified way, for example, a Google Doc that you can fill in anonymously, and then if you need to know, for example, people's age or where they're living in the United States or their gender identity, things like that, you can also ask those questions, but not have it identified in terms of no names, no birth dates, no identifiers, so that you couldn't figure out who filled out the survey. So that's considered ethical. And that's what I would recommend, is look up depression and anxiety scales.
Yeah, it also depends on what you're what your hypothesis is, and what you're trying to measure. So if you have some specific outcome you're trying to measure, maybe you can ask about it in a numerical scale, which makes it easier to do statistical testing, or you may have to ask about it in a different way that might be harder to analyze, but might actually capture whatever you're trying to capture.
Right. Yeah. But I think, so in terms of ethics, I think the big thing is as long as people can answer sort of anonymously and that information is removed from their identity, then that's okay.
Yeah, unless you have some
Um,
type of IRB or something, but you probably don't at this point.
right. Do you think a COVID mutation will delay development of the vaccine? No, the way that the vaccine is being developed is with a very stable protein. And that's why they've been able to use vaccines that they've been working on for MERS, MERS and SARS, to help in the development of this vaccine. So there are different strains of SARS-CoV-2 already, even within the United States.
Yeah, they've done some interesting studies with these because they can tell where the virus likely originated or how it kind of moved through the world because of changes that are preserved. And so they've done studies showing that most of the virus in the United States is coming from Europe and through New York, except for in Washington State, I believe, where a lot of it is coming from China. I haven't followed that closely, though. I think one other thing to consider with the kind of immunity, which we didn't touch on, is the durability of the immunity, and that's how long it lasts. So if you have immunity to COVID and then you're immune for 6 months, that's great for 6 months, but not helpful after that. And I think that's a really big unknown as well, as far as I know, that we, one, don't know if there is a strong immunity, and two, we don't know how long it lasts if it's present. AP research. I've never heard of AP research. That sounds like an interesting thing. What about the MIS-C? Ethan, what do you want to know about the MIS-C? Not guaranteeing that I'll know anything else about it. Yeah, I think that's true. I think there was a kind of big blow up about it, and I think that this is probably, as if you were doing a drug trial, a very rare side effect. But this is a disease instead of a drug. I also have not seen much on the news on it lately. I think it's pretty vanishingly rare. So I think one is it's not well understood, and I think Sage gave a reasonable explanation about sort of what scientists think the origin is. But I don't think that we understand it very well. I don't think we really know how to treat it super well, other than supportive care. And I don't think we have any idea about how to predict which kids might have this response and which may not. So yeah, I think unless Celeste or Sage knows a lot more about it, which it didn't sound like, I think we're in the dark, just as you are. I think this is science. There's a lot of things to learn. This is a new virus, and I think we're still wandering a bit in the dark here.
Yeah, from what I've seen, it kind of falls under the umbrella of because every person's immune system there's wide variation in the immune response between each individual. And for some, for the same antigen, certain people's immune systems might mount a very minimal response, and certain people might mount an insanely high response. And right now, there are various syndromes that kind of follow this pattern, but to this day, we still don't know why one person has a certain response versus another. So from what I've seen, it sounds like MIS-C is one of these reactions that falls under that. Just like how some people, when they get a flu vaccine, they might actually feel like crap after. It's just because their immune system reacted differently to the live attenuated virus. And whereas other people just like don't even get sore on their arm. So this is kind of a reflection of that range of immunogenicity between people. But thankfully, this seems to be quite rare, but it can definitely happen.
Cool. Sorry we can't help more, Ethan, but if I see anything else, I'll keep it in mind. Any other questions? I hope you guys know this comic that I'm presenting here, but I don't think it ran during any of your lifetimes because it ended in 1995. I guess I'll continue with my stuff if we don't have any other questions. You got anything else to say, Sage?
Nope,
Okie
you're
dokes.
good. I
Good to
love
hear.
Calvin and Hobbes.
No, me too. Calvin and Hobbes is a true legend. Okay. This comic is not that related to what I'm talking about, but it has kind of heart things in it. As you know, you will expect to hear about hearts from me. But I'm talking about the large vessel coming off of the heart, which is the focus of my my research, the aorta. I'm going to close the chat right now, but let me know if there's questions. So the aorta, I think you guys are all familiar, and we've talked about it a couple times. The aorta is the large vessel coming off of the left side of the heart. And remember, in radiologic and anatomic images, the left side of the image is talking about the patient's left, which we view on our right. And so the aorta is the main vessel that comes off the heart, and it supplies blood to the rest of the body and even to the heart. And there's a valve there, as you guys know. It's called the aortic valve. And generally, this valve has 3 leaflets. And so there's so when it closes, all 3 of these leaflets come together, and when it opens, they come apart and let blood flow through them. But there's a reasonably common congenital abnormality called bicuspid aortic valve. This occurs in around 1.5% to 2% of the population, and predominantly in males. So maybe 75 to 80% of patients with bicuspid aortic valve are males. And so this means that they only have 2 leaflets in their aortic valve, and you can see here in this image that maybe there's a hint that there should be a third leaflet, but it didn't actually separate, or it's fused there. So there's a raphe, is what it's called, where the leaflet is fused. And we don't actually know what causes bicuspid aortic valve. We think it probably has some genetic factors, but also in my lab particularly, we think that it may have blood flow cause, so maybe blood flow during development is abnormal. And even if it's purely genetic in formation, we think that the blood flow is really important to the disease course. And so some of these patients, as you can imagine, having an abnormal heart valve, it opens and closes, 60 times a minute for your whole life, is going to cause problems down the line. And so the problem that is worrisome from bicuspid aortic valve, they develop valvular complications. So that means the valve doesn't function appropriately, but also the most obvious from a physiologic standpoint is called aortopathy. And this is simply a fancy word meaning dilation of the aorta. You can see in this figure that it can occur. We're talking about, I work mostly in the ascending aorta, though it can occur anywhere in the aorta, a dilation, but generally from the bicuspid valve, we see this in the ascending aorta. And so it can occur near the root, and this can also cause valve complications because it spreads out the area where the valve is, or it can occur anywhere distally to that, so further down the aorta. And so we know from the last couple hundred years of medicine that patients with bicuspid aortic valve tend to have more complications, and it's a large order more complications than patients without a bicuspid, so a normal tricuspid valve. And we know that 50% of bicuspid patients will have complications, and most of these people will progress to surgery at some point in their lives. And there's one complication, aortic rupture. It's the most feared complication. But generally, these patients actually do have some it is noticed because they have some symptom. So they would generally be surgically repaired in the United States before they had an aortic rupture, which is just the blood vessel wall explodes and all the blood would leak out. And that's generally fatal unless you're already sitting in the hospital because you lose so much blood so quickly. And so the treatment here, of course, is surgical replacement. These patients do receive medications, but the evidence is not that great that the medications actually help you reduce the growth of your aorta, or at least they may delay it. And so the guidelines here, and part of the reason that we study this disease is because the guidelines change a lot. In the last 10 years, in the last 15 years, the guidelines have changed more than 10 times. And these are generally based on the size and growth rate of the aorta. You can see here that someone here thinks that there is some correlation between which of the valve leaflets are fused and the shape of the aorta and the dilation that's likely to result from that. So I work with MRI. This is a little disjointed, but I'm not going to explain the physics of MRI in this presentation. I think it's a really easy way to lose people in a presentation. There were 6 Nobel Prizes awarded in the development of MRI, and we're certainly not going to cover all of that in a 20-minute lecture. But essentially, it uses very strong magnetic fields to image the human body. And hydrogen and another few atoms, which are very rarely used, but can be done research-wise, are influenced by this magnetic field. And the magnetic field that we're talking about is around 30,000 times stronger than the Earth's magnetic field. And so this is an incredibly strong superconducting magnet. I'm not sure if any of you guys have happened to had an MRI. I hope not. But essentially, you would lay down on this bed, like this patient is. You would put on hearing protection because they're extremely loud. And then you would be slid into the bore of the scanner. So it's a little bit claustrophobic. And then this gray ring here is the size of the superconducting magnet. So that's cooled with liquid helium. And then there's other magnetic coils in this inner white plastic area, and even these this kind of plastic thing over this guy's legs. This guy's about to have a leg scan. So there's 3 different sets of MRI coils that are used for imaging. And the specific technique that I work on is called 4D flow MRI. And so it's a multi-dimensional technique. So that means that we get a 3-dimensional volume. And this 3-dimensional volume is also time resolved. So we get a movie over the 3D volume. And the interesting part about this technique is that we get the velocity information at each image pixel or each voxel. And so we get a normal image that you see here, which is labeled magnitude in the kind of black and gray. And that shows you what the anatomy looks like. So you can see the heart beating there. You can see the liver is pretty still. And you can see the aorta looking like a candy cane in this image. Then at the same spatial location, so all of these are the same slice of the image. It's only one of the 3-dimensional slices. You can see that we have these other images, which are much harder to interpret. And these represent the velocity at each point. And so we can see, especially on the bottom left, and this is time resolved. So this is showing as the heart beats. You can see that there's a quick point where the velocity kind of goes up, and you can see the aorta, and then it kind of melts back, and the velocity goes down. It's harder to see the aorta. I wouldn't worry too much about how to interpret these here. But this is just one method of measuring flow. The nice thing about it is that it gives us a full 3D volume over time. It has some drawbacks that I'm not going to talk too much about, but there are other methods I just want to mention that we can use to measure flow. Echocardiography or echo ultrasound. This is able to measure flow using the Doppler principle. And then there's some less quantitative measurements using other MRI techniques or even CT or computed tomography. And so a research question that I'm looking at is, can we use information about the anatomy and the blood flow patterns in patients with bicuspid aortic valves to predict who's going to need surgery? And you can see here, this is what an example of our data set looks like afterwards. And this is a unique data set that's looking at about from the head down to the thigh. And you can see that we can see the blood flow, that the vessel that lights up really bright right there is the aorta. And then the other vessels tend to be veins or smaller vessels with much lower velocities. And you can see that there's a pretty nice correspondence between this sort of anatomic diagram on the left, and on the right-hand side, you can see that we can see the blood flow to these organs. So I'll talk about my research now. Obviously, this is a large data set, and data processing is a big part of what I do. And so we take in this data that I showed you. You can see it on the left here. And then there's some pre-processing steps. I wouldn't worry about exactly what they are. But then we can generate this kind of what's called a PCMRA, or a phase contrast MR angiogram. And this is just a fancy way to say that we can see where the blood vessels are. After this, we use this angiogram, so image of the blood vessel, to manually delineate where the aorta is. So we have to kind of draw out in 3 dimensions where the vessel is, so that we can use only the velocity within that vessel, which is the blood flow velocity, for our calculations. And this takes like around 10 minutes if you're really fast, and probably much longer if you're not. And then all in, from getting the data off the scanner to actually visualizing it, can be 15 to 30 minutes, depending on how complicated the patient's anatomy is. And so one of the parts of my research, and this is started as a side project and took over a bit, is doing this segmentation, so telling where the vessel is using artificial intelligence. And so I mentioned a manual observer takes 10 to 15 minutes to do this if they're very good. The artificial intelligence software that we wrote does it in about 1 second. And this is based on convolutional neural networks, which you may have heard about. The downside here is that you need a lot of data to feed into it to teach it how to do these vessels. So you can think of this as, it's not exactly like a human that's learning. Sage already knows what an aorta looks like. And if I asked her to use this software to draw the aorta, she would not need any training. She could do it immediately. And so it learns a little bit differently than a human, but it does learn to be as good as a human, which is surprising to us. So we compared me and someone else to us and the computer is as good as us and many thousand times faster. It also complains a lot less. And so we also implemented something similar that I won't show for the other processing steps, and we got the entire processing pipeline down from around 15 to 20 minutes to under 30 seconds. And it's fully automated with no user interactions. A more current work, and this one doesn't seem to be playing, unfortunately. But and this is the one slide that needs to play. That sucks, too bad. Essentially, what I'm saying here is that most of the time we're using a time average segmentation. This is supposed to show a time resolved segmentation, but happened to be the one GIF that didn't play. So we'll skip it. And one of the metrics that we're interested in with these flow in the aorta is called the wall shear stress. And the wall shear stress, since we're measuring the blood flow velocity, is the force exerted by this flowing blood on the vessel wall, so the aorta wall. And we can calculate this directly from the velocity field that we get from the 4D flow MRI. So we know the velocity at every point, and we can use this to calculate the stresses on the walls of the aorta. And you can see an example of that here. We can look kind of from the front here, and you can see in the red where the wall shear stress is kind of high. And this patient happened to have a jet out of their valve. So the valve is right at the kind of short end of the candy cane. And so the valve here was damaged, and it was causing a jet, and you can see that it caused a really high shear stress pattern on the walls of that aorta. And the reason that the reason that we're into this, I'll talk about it in just a second, but we're able to take individual patients like this. So you might say, well, it looks red, but is that really high compared to normal, or is it low compared to normal, or is it completely normal to have that in this particular patient? And so what I've done in a previous work is we've taken a cohort of age and sex matched controls. So these are patients that don't have any cardiac disease, who are the same age and sex as the patient that we're looking at. And we do some fancy math to average them all together. And we get something that's nice and smooth like this, that shows what the normal shear stress on the wall of the aorta is in that cohort. And then we take an individual patient that we've processed that looks something like this, which in this case, this patient has pretty high wall shear stress. And we can actually compare point by point and kind of push them together and see how they're different. And we can then generate a map that looks like this at the far right, which in red shows where the wall shear stress is much higher than normal, and then in blue shows where it's lower than normal. And what we've used these for is we actually have a study ongoing where we give the surgeons these heat maps when they're resecting these diseased aortas and replacing them with a sort of plastic graft. And people who are much more invested in biology than I am take these tissue samples and they actually look at them under a microscope, and they look at how the structure and how the protein expression changes in these tissues where they were either subjected to very high wall shear stress or in areas of normal wall shear stress. What was shown just before I started in this lab was that in patients with this high wall shear stress, in regions where that wall shear stress was elevated in the aorta, the matrix, the extracellular matrix, that's kind of the structure outside of the cells, is damaged in these patients and degraded. So that may contribute to growth in these patients because the sort of structural components of the aorta wall have been damaged. And then, so that's kind of an overview of kind of the broad strokes of what I do for research. But I wanted to show just one example of like what we see if we're looking at one patient. And so this is a recent case I was asked to look at by some of the surgeons. And this is a patient who's in his late 40s. So it's a man in his late 40s. And he unfortunately has a bicuspid aortic valve. And he needed surgery because his aorta is getting pretty dilated. Right now, the cutoff is around 55 millimeters, sorry, not milliliters. And he also has severe valve disease. So that actually bumps you up and makes it more risky. So they operated on him before he hit this 55 millimeter cutoff. And so surgeons decided to perform the Ross procedure, and I'll describe that in just a second, to repair his aortic valve and his aorta actually. And they asked us to look at the flow before and after to see if his flow had become more normal due to the surgery. And so just a brief aside about the Ross procedure, just because I think it's pretty cool. Essentially, if a patient has a diseased aortic valve, they actually remove that entire aortic valve. So they cut the aorta, they remove the valve, and then they actually cut the pulmonary artery, which is sort of a weird thing that you would not normally think of to solve this problem. And they take out the pulmonary artery and pulmonary valve and put that where the old aortic valve was. So the patient's own pulmonary valve replaces their aortic valve, but now they have no pulmonary valve. And so what they do here is that they take a graft from a donor. So they find a deceased person and they take their pulmonary valve and they actually replace the patient's pulmonary valve with a dead person's pulmonary valve. And the question would be, why would you make a new valve problem to solve an old valve problem? And the reason is because the pulmonary artery of the patient tends to last much longer in the aortic position. They're both 3 leaflet valves. And since the pulmonary artery is much lower pressure than the aortic side is what you measure at blood pressure. It's usually around 140 right at this place. But the pulmonary artery is around 15 to 25, even if it's super high. And so this is done actually pretty commonly in children, even so it creates a new problem, but it tends to solve the aortic valve problem pretty well. And in children, this is a really important treatment because your native valve will still grow. So if you put a fake valve in and it doesn't grow, and then you have to reoperate on these patients. But in this case, this is a pretty early example of doing this in adult patients with this type of disease. And so this is why we're looking specifically at this case. And this will just show you a little bit more what our data looks like. So this patient actually happened to have several 4D flow MRI scans all the way back to 2012. And so they have 8 years of follow-up with this technique. And you can see the wall shear stress starts off a little bit lower and then tends to increase. And then is the furthest right image is after the surgery. And you can see that the shape of the aorta is very different. The patient's aorta is much, much thinner, and that's because it's been replaced with a graft, which is just basically a plastic tube that's sewn instead of the aorta. And these measurements at the bottom, I wouldn't worry too much about them. You can see it's growing, but these measurements are taken at these 2 locations, the sinus of Valsalva, or SOV, and then the mid-ascending aorta, which is marked MAA. And the question that the surgeon asked us was, does the flow normalize? And so if you look at the kind of the left image with the really fat aorta, we notice that there's some pretty complex flow patterns here, and it looks like it's kind of cycling around or helical. Whereas after the surgery, where the aorta is much narrower, you can see that the flow is in nice smooth lines that go smoothly down the entire aorta. And then on the far right, you can see that these, you can see the root of the aorta, and so this is the sinus of Valsalva. And it's important here to have flow go into the coronary arteries, and you can see on the top here, these two little arteries that come off the bulb shape of the aorta. And those are the arteries that feed the heart, so they really need to get blood. And the important flow feature for that is a recirculation that occurs in this bulb due to the shape. And so we show here that this the recirculation is present in this patient after they have their aorta replaced. And so that's really good news. And you can see here that, again, this is a time-resolved acquisition. And so you can see some kind of videos about what this looks like. These aren't perfect, but you can see the heart beating, and you can see the flow in the aorta. And you can certainly see that the flow is much more complicated in the patient on the left, before they have their surgery, and much simpler on the right after they have their surgery.
So
we have a question in the chat. So the question is, does it specifically have to be a homograft for replacement pulmonary valve?
Sorry, can you repeat that? I couldn't hear you.
Does it specifically have to be a homograft for the pulmonary valve?
For the Ross procedure?
Yeah.
I don't actually know. I think it generally is. I mean, in theory, you could use the aortic valve to replace the pulmonary valve, but I think that it's almost always a homograft, and it's the same valve. You could use a mechanical valve in this case to replace the pulmonary valve, but I think that has a different name, or is much more rarely done. I'm not sure there.
Don't some people use bovine or porcine valves?
Yeah, these are certainly used. So yeah, I say mechanical valve very, very generously, but generally the valves that are used to replace these valves are either, I guess, mechanical, as I said, and this is what you think of if they're made of steel and or kind of carbon compounds. And if they're made out of biologic tissues, then they would be kind of a bioprosthetic valve. Certainly those are used as well. Yeah, that's a good question. You know a lot more about heart valves than I would have expected. Yeah, and that's what I had to talk about today. We certainly believe that hemodynamics or blood flow plays an important role in the development of a dilated aorta, particularly in patients with valve disease. And so that's what I've been focusing on. Science is slow. I mean, you're seeing a pretty broad overview, but this is 3 years of work and much more work than that on the part of collaborators and other people who have helped me out on this project. And so, yeah, I'll put an acknowledgement slide in there, but any questions, I'm happy to answer. I had a couple good questions already. But yeah, as you can see here, this is a pretty collaborative effort. There's probably 25 people on the slide, all of whom have contributed in some way to these projects. And so it's kind of a cool project to be able to work on with so many people doing this kind of stuff. Let's see, I'll stop sharing. Oh yes, okay, so we had, is there a preventative measure that can be taken? Yeah, there is a preventative measure that can be taken, but I think the evidence is not so good. Essentially, there's a disease called Marfan's disease, which is a connective tissue disease. And patients with this type of disease tend to have extremely dilated aortas. Their tissue tends to stretch very easily. And so they have these ridiculously huge aortas compared to normal people. And so a lot of the evidence for treatment of patients with dilated aortas comes from these patients. And the physiology is very different. Or the pathophysiology is very different. So it's not clear that using the same treatment in a patient with normal tissue compared to Marfan disease is actually the right treatment. And so generally these patients that have really large aortas will be placed on beta blockers. So these are drugs that can help lower the blood pressure and the force of contraction that the heart contracts with. And they're very safe drugs. They're used very widely. But most of the evidence here is from Marfan's disease, and we're actually writing a paper now showing in a large cohort that we've looked at. Actually, it's not that large, but we've been able to show in our patients at least that having a beta blocker did not help them slow their aortic growth. And that's obviously just a retrospective study where we didn't randomize the groups or anything like that, so it's not perfect. But we're suggesting that maybe beta blockers are not the treatment of choice. We've also recently had work looking at some other drugs which may modify tissue properties, or blood pressure as well. So this is angiotensin receptor blockers, so ARBs. And so we've shown that patients here may have some beneficial properties compared to doing no treatment at all. And so, yeah, there are preventative measures. I think we're still exploring what the best way to treat these patients are. It's a little bit difficult because the progression is extremely slow. So these patients grow, your aorta is growing about 1 millimeter per decade diameter. And these patients may grow 10 times that fast, but that would be really fast growth. And so even resolving the growth change, the resolution of our MRI is around 2 millimeters per voxel. And so we need them to grow more than that to be able to see the changes. And so even that has been a challenge so far. And now that we're at about 7 to 8 years of follow-up on some of these patients, we're starting to see it definitively, but it's been a really long time just waiting for data to come in. So yeah, that's what I have. My work is very different than the others, but I think it's kind of nice to see that there are some non-wet lab biology things that you can do. I'm by no means a biologist anymore, so I spend most of my day programming.
Cool. Thanks for sharing, Mike.
Couldn't have Prism without me talking for 45 minutes or something, right?
It's true. A staple. As usual, if anyone has any suggestions for next week, please let us know.
And I know we've had some suggestion. That's good. TED Talk.
TED Talk.
I'll take a look. I know we had some requests for anatomy and physiology, so we'll try to throw some anatomy and physiology in. If you have any particular organ systems you'd like to hear about, please tell us. If you want to have deep sea physiology, you can do that. No, I'm just kidding. Organic chemistry. Oh, wow. Okay. Well, we could do something on organic chemistry.
Get a guest speaker on that one.
Yeah, maybe. Interested in the electrical activity of the heart and reading an EKG in depth. That is going to be a tall order. I think we can certainly talk about an EKG, but I don't know if we're going to go into too much depth about exactly how to read it. And part of the reason is because we have been trained to do this for a week or so, and that is not nearly enough to get and not nearly enough to be experts and be teaching you guys. So we can do something about that. Is it difficult to get a donor's pulmonary artery? That's a very good question. The answer is probably, but the nice thing about pulmonary arteries, and they have done a lot of different studies on this, is they used to kind of like chemically treat them to try to make them less likely to elicit an immune response because they're a transplanted organ. But they've really tuned that in, and they don't have so many immune response problems with them anymore. But the other thing that they discovered is that they freeze really well. So they generally freeze them, and then they have cancer. We do have some cancer specialists. At least we got one cancer specialist here, maybe blood cancer at least. But yeah, the pulmonary arteries valve, so the pulmonary valve tends to freeze pretty well. And so they generally are using frozen or preserved arteries here. If we did a unit on cancer, I think cancer would be an interesting one. Cancer is very broad.
There are some underlying themes, especially in the molecular biology. So we can definitely talk about that. I'll try not to get too much into the biochem.
Sounds like we want some biochem.
Well, I was thinking for organic chem, we could potentially get a guest speaker.
Yeah,
it's definitely
Dalton.
not in our wheelhouse. Yeah.
Potentially, yeah, certainly. So one of the guys in our class is I don't know if this is something that you're interested in, but he's a synthetic chemist, and he basically synthesizes drugs. At least that's what I understand that he does. So we could potentially, would you guys be interested in hearing his research? We might be able to get him on the line.
He's potentially making
in terms of
inhibitor of cancer proliferation.
Oh yes, Dalton has been requested. Oh nice. Okay.
In terms of cancer, is there a specific cancer type that you're interested in? Because we could get, I mean, I can think of 5 people in my cohort that study cancer, but they all do quite different.
Yeah, but we could always start with a kind of general overview of like cancer principles.
Yeah.
But certainly, yeah.
But I just think if someone has a particular interest, then we can target like a, yeah.
I'm just interested in not having it. But, okay. Cancer, what else do we have? Cancer, organic chemistry, all right. That's not my bag of tricks. There's a TED Talk. Okay, breast or prostate? Okay. Strangely, I am published in prostate. Am I published in pancreatic cancer? I don't remember.
You're in pancreatic, Mike. I've heard that story.
Pancreatic. I always forget. So I will not be the person lecturing on this. I can't even remember the kind of cancer that I was published on.
Your own work? Come on.
Yeah, I'm like 6th author. I did the mechanical stuff.
Oh,
I worked in a breast cancer lab once upon a time, a long, long time ago, in another country.
I mean, I can definitely talk more about leukemia as like the disease itself instead of a specific, very, very specific molecular biology aspect of it.
Okay.
So we can do that.
Cool. So it sounds like cancer. We'll see if we can get some of our friends to either talk about cancer or about any topic on biomedical science or engineering would be great too. Okay. Sounds good. If you have anything specific you want, certainly bring that up. I mean, I know a lot about medical imaging. That's my real expertise. I'm not an expert in tissue engineering, but it's an interesting field. I don't think Stephen is either, though. He's not BME, is he? No.
No, yeah. Stephen's not BME. Andreas is BME.
Yeah, but he does a lot of stuff like me.
He does wearables, yeah.
I think Malika's in
Shuan is BME. She's at the NIH doing drug delivery and cancer. Yeah.
Yeah, there's a lot of drug delivery work at Northwestern, that's true. And at the NIH as well. Okay, well, we'll think about it. Thanks for the suggestions. Do you have anything, Blanca?
I think that, one more. Drug delivery sounds interesting.
Oh, I set myself up for that. All right. We'll see who we can get. We'll ask some of our friends and see who's got time to present. Certainly they're doing a lot of cool stuff.
Okay, but I think besides that, it was a really great class. I want to applaud all the students who took time to like be here and also ask a bunch of amazing questions. It made this entire process really awesome for us, you know,
Yeah,
especially for
it's
the
much
mentors.
more fun than talking into the void. So thanks for participating, guys.
Yes.
So thank
Yeah,
you
this is
guys.
great.
We'll see you next week. Thanks.
You guys
And have,
Thanks, everyone.
and
enjoy
have
the
a
rain.
great
Enjoy the rain.
graduation for everyone who's graduating.
Yeah, good luck with finals if you have those.
Yeah.
Sorry. Very enriching. There we go.